8.1 Origin of life. Theory of spontaneous generation, abiogenesis. Abiogenesis is the generation of life from non-living matter. Abiogenesis, now more precisely known as spontaneous generation. This theory that complex living organisms are generated from decaying organic substances, e.g., organism like mice spontaneously appear in stored grain or maggots spontaneously appear in meat. Francesco Redi, an Italian physician, was the first to disprove the theory of spontaneous generation by performing a controlled experiment. In 1668, Francesco Redi performed an experiment to check whether maggots really came from decaying meat. He did this by placing meat in a number of jars and covering half of them with fine gauze while leaving the others uncovered. Maggots developed only on the meat in the uncovered jars. Hence, Redi concluded that the maggots did not come from the meat, but from tiny eggs that flies had laid on the meat. Since flies could not land on the meat in the covered jars, they could not lay eggs on that meat, and no maggots formed. Therefore, decaying meat could not produce maggotae. Later, Lazzaro Spallanzani, an Italian naturalist, performed a similar experiment with broth. He put broth into 2 glass flasks and sterilized them by boiling the flask containing broth. One of the flasks was left open to the air. The other flask was sealed up to keep out any organisms that might be floating in the air. Microorganisms developed only in the uncut flask. From this, Spallanzani concluded that the microorganisms did not come from the broth, but were in the air that entered the flask. Broth heated flask left open growth. Figure 8.1. Sterilized broth in open flask. Developed microbes whereas in sealed flask microbes not developed. Broth heated flask sealed no growth. Unfortunately, many scientists were not convinced by his experiment. Louis Pasteur, a French chemist who finally disproved the theory of spontaneous generation in the mid-1800s. He performed the same type of experiment as Spallanzani. Louis Pasteur, however, allowed air to enter into the flask of sterile broth. He performed experiment with 2 flasks, one with straight neck and other with S-shaped neck. Flask with a straight neck allowed both air and microorganisms to enter, whereas the other flask with S-shaped neck allowed only air to enter but not microorganisms. The straight neck flask became contaminated with microorganisms, but the broth in the flask with a S-shaped neck did not become contaminated. Therefore, Louis Pasteur showed that even though air could get in the flask, the broth did not produce microorganisms. 437. Weight. No growth. Figure 8.2. Nay microbial growth. Occurs in flask with a S-shaped neck whereas microbes growth occurs. Boyle break stem * microbial growth in flask with broken stem. Scientists finally were convinced that living things, no matter how small, do not come from non-living things. The present theory of where living things come from is called biogenesis. This theory states that living things come only from other living things. For example, mice come only from mice, and microorganisms such as bacteria can only come from other bacteria. Since spontaneous generation was now proved incorrect, many scientists began to wonder how life started on Earth. The Earth formed about 4.5 billion years ago. It is very difficult to determine life's origin because it began at least 3.5 billion years ago. But the preparation for life had to begin a long time before, i.e., some 4.3 billion years ago. Years after, under the influence of internal terrestrial heat and solar radiation, the first molecule capable of reproducing themselves in favor catalytic reactions emerged. The crucial event could have occurred 3.8 billion years ago, but, in which condition was this prebiotic soup cooked? A. I. Apparent attempted to answer this question. Chemical evolution. The generation debate heated up again in 1924 when Russian biochemist A. I. Apparent proposed that life had arisen from simpler molecules on the lifeless Earth under much different atmospheric conditions than exist today. How, instead of life arising subtly, as previous spontaneous generation theories proposed, Apparent believed that it occurred over a very long period. However, instead of life arising subtly, as previous spontaneous generation theories proposed, Apparent believed that it occurred over a very long period of time. Apparent and Haldane, working independently, proposed a theoretical model for chemical evolution. The Apparent suggested that under the strong reducing conditions, theorized to have been present in the atmosphere of the early Earth between 4.0–3.5 billion years ago, inorganic molecules would spontaneously form organic molecules, simple sugars and amino acids. English biologist J.B.S. Haldane published a paper in which he proposed that ultraviolet light, acting on a primitive reducing atmosphere containing water, ammonia and methane produced oceans with the consistency of a hot dilute soup containing the building blocks of life. Haldane's theory is most famous for its reference to the hot dilute soup of the early Earth. In 1930, Stanley Miller, along with his graduate advisor Harold Urey, tested this hypothesis by constructing an apparatus that simulated the apparent Haldane, early Earth Miller-Urey experiment. The experiment, a classic experiment, fundamentally established that Earth's primitive atmosphere was capable of producing the building blocks of life from inorganic materials. It established that the conditions that existed in Earth's primitive atmosphere were sufficient to produce organic molecules like amino acids. 438. Evolution. Tungsten. 5-liter flask. Figure 8.3. Apparatus for simulating the synthesis of organic compounds on the prebiotic Earth. A mixture of gases thought to resemble the primitive Earth's reducing atmosphere is subjected to an electric discharge to stimulate the effects of lightning, while the water in the flask is refluxed so that the newly formed compounds dissolve in the water and accumulate in the flask. Condenser. A.R. In 1950, University of Chicago researchers Stanley L. Miller and Harold C. Urey set up an experimental investigation into the molecular origins of life. Their innovative experimental design consisted of the introduction of the molecules thought to exist in early Earth's primitive atmosphere into a closed chamber. Methane, CH, hydrogen, H, ammonia, NH, gases were introduced into a moist environment above a water-containing flask. To simulate primitive lightning discharges, Miller supplied the system with electrical current. After a phase, Miller observed that the flask contained organic compounds and that some of these compounds were the amino acids that serve as the essential building blocks of protein. Using chromatological analysis into his experimental observations and confirmed the formation of amino acid section glycine, alanine, aspartic acid, glutamic acid, beta-alanine hydroxy acids, and other organic compounds alkyurea, lactic acid, glycolic acid, succinic acid, proprom C acid. Thus, the experiment demonstrated how some biological molecules, such as simple amino acids, could have arisen abiotically, that is through non-biological processes, under conditions thought to be similar to those of the early Earth. Condensation and polymerization. Miller and Urey demonstrated the formation of simple biomolecules. The chemical evolution would depend on the polymerization or condensation of these monomer into polymer, i.e., formation of macromolecules like polypeptides, polysaccharides. For example, nucleic acid bases can be synthesized under supposed prebiotic conditions. In particular, adenine is formed by the condensation of HCN, a plentiful component of the prebiotic atmosphere, in a reaction catalyzed by NH. The other bases have been synthesized by similar reactions involving HCN and H0. Sugars have been synthesized by the polymerization of formaldehyde, CH0, in reactions catalyzed by divalent cations, alumina, or clays. It is probably no accident that these compounds are the basic components of biological molecules. They were apparently the most common organic substances in prebiotic times. Proteinoids, thermal proteins. In the 1950s, Sidney Fox and co-workers, University of Miami, produced synthetic peptide-like products of molecular mass between 4,000 and 10,000 Daltons by heating a dry mixture of amino acids at 150 degrees Celsius to 439 FUIULIUIN 80 degrees Celsius, which he called proteinoids. Unlike the linear proteins produced by cells, proteinoids are branched polypeptide chains. Exhibit several properties similar to biological proteins, including catalytic activity and susceptibility to digestion by protease enzymes such as pepsin and trypsin. Region of organized structure I. Complex molecules were produced from simpler ones, how might they have become organized into cells? Macromolecules aggregate into membrane-enclosed droplets, conservatiges and microspheres, as a result of intermolecular forces, which exhibit some features of living systems such as organization, selective permeability, and energy use. OASERVATES. The term introduced by Apparin is an aggregation of colloic Al particles in liquid phase that persists for a period of time as suspended membrane droplets. Formation of conservates occur when dispersed alloidal particles separate spontaneously out of solution into droplets because of special condition of acidity, temperature and so on. Conservates show a number of interesting properties, able to exchange substance with environment. Can increase in size. Selectively concentrate compounds within them. ICROSPHERES molecular aggregates of proteinoids. Showed that microscopic membrane-bound spheres, termed microsphere, formed when proteinoids reboiled in water and allowed to cool. Microspheres are uniform II in size, stable in double membrane and _T appear somewhat cell-like, and can undergo fission and T-udding. Some cell-like properties, UCH's osmosis, growth in size, and selective absorption of chemicals have been ascribed to them. Self-assembly of macromolecules into coacervates, and microspheres indicates that the occurrence of similar structure under primitive conditions would probably give rise to more organized structure, protocells. Protocells that could reproduce would be acted on by natural selection, which rolled act as a creative force successively enhancing the probability of advantageous molecules, reaction and destructure. Lembranes defined the first cell. One of the crucial events leading to the formation of the first cell must have been the development of an uter membrane. The need for is easily fulfilled by a class of molecule that has the simple physicochemical property of being amphipathic, that is, consisting of one part that is hydrophobic, water W soluble, and another part that is hydrophilic, water soluble. When such molecules are placed in water, aggregate, arranging their hydrophobic portions as much in contact with one another as possible and their hydrophilic portions in contact with the water. Amphipathic molecules of appropriate shape spontaneously aggregate to form bilayers, creating small closed vesicles whose aqueous contents are solated from the external medium. All present-day cells are surrounded by a plasma membrane consisting of amphipathic molecules, mainly phospholipids. The first membrane-bound cells were formed by spontaneous assembly of phospholipid molecules from the prebiotic soup, enclosing a self-replicating mixture of RNA and other molecules. It is not clear at what point in the evolution of biological catalysts and protein synthesis this first occurred. In any age, RNA molecules were sealed within a closed membrane. They could begin to evolve in earnest as areas of genetic instructions. They could be selected not merely on the basis of their own structure, but also according to their effect on other molecules in the same compartment. 140. Evolution. Gases of the primitive Earth atmosphere. So, NH, CH, HCN, HO. Heat or other forms of energy plus water. Amino acids. Heat anhydrous conditions. Dot. E. E. S. Dash. Closing square bracket. Proteinoids. Water. Protocellus. B. Mild heat. Replication of protocells. Energy storage retrieval mechanism, ATP. Photosynthetic. Internal protein mechanism, synthesis. Nucleic acid synthesis, e-heteromate. Protoribosomes. Genetic coding mechanism. Contemporary cells. Figure 8.4. Figure 8.4, outline of the major steps postulated for the origin of modern cells. Current models. There is no true, standard model, for the origin of life. But most currently accepted models build in one way or another upon a number of discoveries about the origin of molecular and cellular components for life, which are listed in a rough order of postulated emergence. The prebiotic conditions result in the creation of certain basic small molecules, monomers, of life, such as amino acids demonstrated in the Miller-Urey experiment by Stanley L. Miller and Harold C. Urey. Plus phospholipids can spontaneously form lipid bilayers, a basic component of the cell membrane. The polymerization of nucleotides into random RNA molecules might have resulted in self-replicating ribozymes, RNA world hypothesis. Pressures for catalytic efficiency and diversity result in ribozymes which catalyze peptidal transfer, hence formation of small proteins, since oligopeptides complex with RNA to form better catalysts. Thus the first ribosome is born, and protein synthesis becomes more prevalent. Propanes outcompete ribozymes in catalytic ability, and therefore become the dominant biopolymer. Nucleic acids are restricted to predominantly genomic use. AA1. Evolution. Evolution of the cell. The earliest cells probably arose about 3.5 billion years ago in the rich mixture of organic compounds, the primordial soup, of prebiotic times. They were almost certainly chemoheterotrophs. Acquired the capability to derive energy from certain compounds in their environment and to use that energy to synthesize more and more of their own precursor molecules, thereby becoming less dependent on outside sources of these molecules, less extremely heterotrophic. Very significant evolutionary event was the development of photosynthetic ability to fix Colorado into more complex organic compounds. The original electron donor for these photosynthetic organisms was probably H(S), yielding elemental sulfur as the byproduct, but at some point cells developed the enzymatic capacity to use H(0) as the electron donor in photosynthetic reactions, producing O. The cyanobacteria are the modern descendants of these early photosynthetic O producers. One evolutionary road occurred when there was a transition from small cells with relatively simple internal structures, the so-called prokaryotic cells, which include the various types of bacteria, to a flourishing of larger and radically more complex eukaryotic cells such as are found in higher animals and plants. Fossil records show that earliest eukaryotic cells evolved about 1.5 billion years ago. Details of the evolution from prokaryotes to eukaryotes cannot be deduced from the fossil record alone, but morphological and biochemical comparison of modern organisms has suggested a reasonable sequence of events consistent with the fossil evidence. Three major changes must have occurred as prokaryotes gave rise to eukaryotes. First, cells acquired more DNA. Mechanisms evolved to fold it compactly into discrete complexes with specific proteins and to divide it equally between daughter cells at cell division. These DNA protein complexes called chromosomes become especially compact at the time of cell division. Second, as cells became larger and intracellular membrane organelles developed. Eukaryotic cells have a nucleus which contains most of the cell's DNA. Enclosed by a double layer of membrane, the DNA is thereby kept in a compartment separate from the rest of the contents of the cell, the cytoplasm, where most of the cell's metabolic reactions occur. Finally, eukaryotic cells, which were incapable of photosynthesis or of aerobic metabolism, pooled their assets with those of aerobic bacteria or photosynthetic bacteria to form symbiotic associations that became permanent. Some aerobic bacteria evolved into the mitochondria of modern eukaryotes, and some photosynthetic cyanobacteria became the chloroplasts of modern plant cells. 8.2they_ORIESOE Evolution. What is evolution? Biological evolution, simply, is descent with modification. Evolution is technically defined as a gradual process in which something changes into a different and usually more complex or better form. As is most famously used, evolution is the process by which an organism becomes more sophisticated over time and in response to its environment. Evolution helps us to understand the history of life. Misconception: evolution is a theory about the origin of life. Evolutionary theory deals mainly with how life changed after its origin. Let's try to investigate how life started, e.g., whether or not it happened near a deep-sea vent, which organic molecules came first, etc., but these considerations are not the central focus of evolutionary theory. How life started, afterwards it branched and diversified, and most studies of evolution are focused on those processes. 442. Evolution. Lamarckism. Jean-Baptiste Lamarck, French biologist, published the first truly mechanistic theory of evolution in his book Philosophie zoologique called The Theory of Use and Disuse of the Inheritance of Acquired Characteristics according to this theory. 1. The ecological conditions in which an organism lives causes it to have certain needs. 2. These needs are met by modification of old organs or by production of new rudimentary organs. 3. By continual use of these rudimentary structures, they increase in size and functional capacity, and by disuse they may degenerate and become lost. 4. Character brought by the environment during the life of an individual become hereditary and thus can be transmitted to the next generation. A. Driven by an inner need, B. The modified heredity, C. After many generations of a short ancestor of the ancestor is such behavior. Descendants stretches to feed on leaves manifested by slightly all have much longer necks. High in a tree, the use of longer necks in its. The neck changes the animal's offspring. Heredity according IY. Theory 8.5. Example of the evolutionary Theory 8.5. Example of the evolutionary concept of acquired characteristics. Lamarck would say that cave fish are often blind because their ancestors had lived for several generations in dark conditions, and in each generation the eyes had progressively degenerated through lack of use. The long neck of the modern giraffe could also be explained by the Lamarckian theory. During times when food was scarce, the short-necked ancestors needed to stretch their necks to get leaves higher up in the trees. In this way they stretched their necks. In generation, the exercising of the neck in this fashion caused an increment in the length of the neck, which become genetically fixed. Lamarck's theory may sound plausible, but it is unsupported by any unequivocal scientific evidence. Darwinism. Charles Robert Darwin, the 12th of February 1809 to the 19th of April 1882, was an English naturalist who proposed and provided evidence for the scientific theory that all species have evolved over time from one or a few common ancestors through the process of natural selection. This became widely accepted by the scientific community in the 1930s, and now forms the basis of modern evolution theory. 443. As solution. Darwin compiled evidences for evolution by natural selection for about 20 years. Between 1842 and 1844 Ney wrote a 230-page essay summarizing his theory and the evidences for it. In the 1850s, he began working on a detailed, multi-volume book to present his theory to the scientific community. Darwin might never have sent completed the book if another British scientist, Alfred Russel Wallace 1823–1913, had not come up with the same idea in 1858. Wallace formulated his theory and wrote it in an essay, which he sent to Darwin. Darwin's fellow scientists persuaded him to let them present his theory and Wallace's essay jointly at a scientific meeting. The presentation excited very little attention, according to the modest Darwin. However, the publication of Darwin's book The Origin of Species in 1859 changed biology forever. Darwin clearly and logically presented the idea that natural selection is a mechanism of evolution. In Darwin's own lifetime, many scientists became convinced that evolution occurs by means of natural selection. Today this theory is the unifying one for all biology. Darwin's theory of evolution by natural selection. First, Darwin stated that variation exists among individuals of a species. Second, stated that scarcity of resources and a burgeoning population would lead to competition between individuals of the same species because all use the same limited resources, struggle for existence. Such competition would lead to the death of some individuals, while others would survive. From this reasoning, Darwin concluded that individuals having advantageous variations are more likely to survive and reproduce than those without the advantageous variations, survival of fittest. Table 1. The logical arguments for natural selection. Fact 1. All species have such approximately 8 great potential fertility that their population size would increase exponentially if all individuals born would again reproduce successfully. Fact 4. No two individuals are exactly the same, rather, every population displays enormous variability. Fact 2. Except for minor annual fluctuations and occasional major fluctuations, populations normally display stability. Inference 3. Over the generations, this process of natural selection will lead to a continuing gradual change of populations, that is, evolution, and to the production of new species. Fact 3. Natural resources are fact 5. Much of this variation limit in a stable environment is heritable. They remain relatively constant. E. 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 M. E. Two. Survival in the. It means that there must. Colon dot. Colon dot. Evolution. A. Heritable variation in neck. B. During times of food scarcity. C. After many generations. Colon dot. Evolution. In this way, according to Darwin, evolution by natural selection would occur. In this way, Darwin coined the term natural selection to describe the process by which organisms with favorable variation survive and reproduce at a higher rate. An inherited variation that increases an organism's chance of survival in a particular environment is called an adaptation. Over many generations, an adaptation could spread throughout the entire species. In this way, according to Darwin, evolution by natural selection would occur. Colon dot. Evolution. A. Heritable variation in neck. B. During times of food scarcity. C. After many generations. Length exists in the ancestral, the longer-necked individuals of this selective process. Population their kind in much greater. Summary. Darwin's theory invariably had a combination of some of the following 5 theories in mind. 1. Evolution is such. This theory states that the world is not constant or recently created nor perpetually cycling, but rather is steadily changing, and that organisms are transformed in time. 2. This theory considers that every group of organisms descended from a common ancestor, and that all groups of organisms, including animals, plants, and microorganisms, ultimately go back to a single origin of life on Earth. 3. Multiplication of species. This theory explains the origin of the enormous organic diversity. It postulates that species multiply, either by splitting into daughter species or by budding, that is, by the establishment of geographically isolated founder populations that evolve into new species. 4. Gradualism. According to this theory, evolutionary change takes place through the gradual change of populations and not by the sudden, saltational production of new individuals that represent a new type. 5. Natural selection. According to this theory, evolutionary change comes about through the abundant production of genetic variation in every generation. The relative individuals who survive, owing to a particularly well-adapted combination of inheritable characters, give rise to the next generation. Neo-Darwinism. Darwin proposed his theory of evolution by natural selection without knowledge of the sources of variation in populations. After were rediscovered and genetic variation was shown to be generated by mutation, Darwinism and Mendelism were used as framework of what came to be called the synthetic theory of evolution or Neo-Darwinism. According to this theory, although mutation is recognized as the ultimate source of genetic variation, natural i.e., positive selection is given the dominant role in shaping the genetic makeup of populations and in the process of gene substitution. Evolution. 8.3 Evidences of evolution. Categories of evidences supporting evolution. Evidences supporting the evolution model comes from multiple, distinct areas of biology and geology. These pieces of evidences can be grouped into 4 categories. 1. Direct observation of evolutionary change. 2. Homology and development. 3. Vestigial traits. 4. Fossil record and biogeography. 1. Direct observation. Small-scale evolution can be observed in nature or generated experimentally in the laboratory. The classic story of the peppered moth in Britain during the Industrial Revolution is an example of microevolution. Grant have recorded evolutionary change in beak shape within and among populations of Darwin's finches, over periods as short as 2 years. We can all drive genetic change in laboratory populations of Drosophila and other model organisms in the laboratory. By phenotypic traits, we can generate directional change in characteristics such as abdominal bristle number, lifespan, and avoidance of certain chemicals. Artificial selection experiments are also the foundation of agricultural improvement over the past 10,000 years. 2. Homology and development. The most compelling lines of evidence supporting the common ancestry of species is that of similar structural elements across functionally diverse forms. It becomes evident from comparative anatomy. We find the same bones in many different types of animals, but these bones are often modified to do different things. The hopping legs of the frog contains the same bones as our own legs, but the frog's legs are highly modified to fulfill a different function, hopping. The wing of a forelimb of a bat contain exactly the same bones as the arm of a human, but the size, shape, and even internal structure of these bones are all adapted to play a different role in each animal. We call structures like the wings of a bird and the foreliages of a bat homologous structures. Homologous structures are structures that are derived from a common ancestor. Even if they are superficially different, they are developmentally related. Homology does not mean that these structures must share the same function. L. Human, frog, bat, porpoise, horse. Figure 8, 7, homologies between the forelimbs of 4 mammals and a frog, showing the ways in which the proportions of the bones have changed in relation to the particular way of life of the organism. Evolution. But very often in nature we find structures that are superficially similar, even though the organisms are completely unrelated to one another. These structures may even serve the same function, like flying, and described as analogous structures. The wing of a bird and the wing of an insect are good examples of analogous structures. In every physical and biological way, these wings are radically different from one another. One is a flat plane of exoskeletal material, the other is a chordate forelimb shaped into an airfoil, with hollow internal bones and an outer covering of feathers. But they can both be used to fly. Observations of structural homology support the causal hypothesis of evolutionary homology, that common structural organization results from common ancestry. The evidence of analogous structures further support the evolutionary homology concept, as they indicate that design is not necessarily constrained, that is, there's more than one way to build a functional part. In many cases, during embryonic development of organism, the embryo exhibits characteristics of the embryos of its ancestors. For example, early in their development, human embryos possess gill slits like a fish, and later exhibit a tail, the vestige of which phased in adulthood as a coccyx at the end of spine. These relic developmental forms suggest strongly that our development has evolved. 3. Vestigial structures. Many species show structural or genetic features that serve no apparent function. Examples of these include the presence of pelvic bones in snakes and whales, rudimentary eyes in some cavefish species, presence of nictitating membrane in human, and the presence of pseudogenes throughout the genomes of eukaryotes. These structures suggest strongly that the species has changed over time, and descended from an ancestor with different ecological or genetic characteristics. Suggesting descent with modification is evidence of poor engineering or inefficient design in some characteristics of organisms. For example, gene regulation is often a very inefficient process with multiple, often conflicting, stages. These objects suggest that existing structures are often coopted during evolutionary change to serve new functions, that is, there is descent, with modification as opposed to independent design of each species. 4. The fossil record and biogeography. In the 1900s, the modern synthesis unified the CONI, EPTS of Darwinian evolution with the principles of Mendelian genetics and the observable patterns in the fossil record. In particular, George Gaylord Simpson demonstrated that the fossil record was consistent with an evolutionary model of the history of IIFE. The support for evolution in geology comes in several forms. First, we can observe the existence of transitional forms between major groups of organisms. These include Geopteryx and other bird-like reptiles, and the therapsid reptiles, which suggest the transition between reptiles and mammals. In addition, the layers of fossil strata are concordant with these evolutionary transitions. Finally, biogeographic distribution of extinct and extant forms is consistent with our understanding of both plate tectonics and Vicarian events, as well as our estimates of the genetic relationships of species. Biogenetic law. Ernst 1834–1919 studied embryology in his dictum that ontogeny recapitulates phylogeny, called the biogenetic law, also called the theory of recapitulation. Ontogeny is the embryological development process of a species, phylogeny is the evolutionary history of a species. ESPD used the view that evolution generally proceeds by placing each innovation on top of a previous one, like adding layers on a cake. Therefore, the embryo of an advanced organism should pass through, recapitulate, the adult stages of more primitive forms as it develops. However, repeated observations of development by other workers, e.g., Wilhelm His, Walter Garstang, Wilhelm Roux, Adam Sedgwick. Evolution. Vendayer clearly showed that embryos do not go through ADUIT stages of lower forms, rather, they share many common features in development. For example, the human embryo at one point seems to have gill slits in the neck, thought by Haeckel to signify a fish-like stage in development. However, gill slits are not gills, and the similarity is not an actuality. In reality, the gill slit develops into the lower jaw in mammals. 8.4 Natural selection. Selection. Tilde. Composite of all the forces that cause differential survival and differential reproduction among genetic variants. When the selective agencies are primarily those of human choice, the process is called artificial selection. When the selective agencies are not those of human choice, it is called natural selection. Evolution as a biological mechanism is driven by natural selection. This theory is favored by many scientists to explain phenomena in nature, so much so that it is generally assumed as factual in most studies. Natural selection is the machine that drives evolution. This mechanism causes those organisms that are abnormal to survive an environmental change, making them the new normal. Over time, according to the theory of evolution, this can cause an organism to change into a totally different form of life. Some evidence of natural selection has been seen in nature, but not to an extent that would change a species in any meaningful way. Every genetic mutation that science has observed changing the form or function of an organism has resulted in handicap or death. This, however, mean that an ecosystem is vulnerable to rapid change, since organisms that cannot adapt will usually die. Evidences of natural selection. Industrial melanism. Industrial melanism is a phenomenon used to describe the evolutionary process in which initially light-colored organisms' population became dark as a result of natural selection. Industrial melanism affected over 70 species of moths in England. It has been best studied in the peppered moth, Biston beetleria. Prior to 1800, the typical moth species had a light color pattern. Dark-colored melanic moths were rare. During the Industrial Revolution, other industrial wastes darkened tree trunks and killed off lichens. The light-colored moth became rare and the dark-colored moth became abundant. By 1886, dark-colored melanic were far more common, illustrating rapid evolutionary change. The cause of this change to be selective predation by birds, which favored camouflage coloration in the moth. Darwin's finches. Darwin's finches are a group of 14 species of small birds, 13 of which occur on the Galapagos Islands. This group of birds provides one of the most striking and best studied example of evolution on islands. The Galapagos Islands are a particularly striking natural laboratory of evolution. These are all relatively young in geological terms, and they have never been connected with the adjacent mainland of South America or with any other source area. All the organisms that occur on these islands have reached there by crossing the sea, as a result of chance dispersal in the water, by wind, or by transport via another organism. Since the Galapagos are a group of islands, small flocks of birds could occasionally reach new islands where no other land birds were present. On these new islands the immigrants would become adjusted to the local conditions through the process of natural selection. In doing so, they would have become progressively more different from the original populations. Descendants of the original finches that reached the Galapagos Islands now occupy many different kinds of habitats on the islands. These habitats encompass a variety of niches comparable to those occupied by several distinct groups of birds on the mainland. Among the 13 species of Darwin's finches that inhabit the Galapagos, there are 3 main groups. The 3 groups are 1. Ground finches. There are 6 species of ground finches. Most of the ground finches feed on seeds of different sizes. The size of their bills is related to the size of the seeds on which the birds feed. 2. Tree finches. There are 6 species of tree finches, belonging to several diverse groups. These species also differ from one another primarily in bill size and shape, which reflect adaptations to their food. 4 trees have bills that are suitable for feeding on insects. The ones with larger bills feed on larger insects, and those 4 with smaller bills feed on smaller insects. One of the tree finches have parrot-like beak. It feeds on buds and fruits and trees. The 6th species of tree finch, the woodpecker finch, has a chisel-like beak. This unusual bird carries around a twig or a cactus spine, which it uses to probe for insects in deep crevices. It is an extraordinary example of a bird that uses a tool. 3. Warbler. This unusual bird differs from the ground finches and the tree finches in its habits and morphologic adaptations. The warbler plays the same ecological role in the Galapagos woods that warblers play on the mainland, searching continually over the leaves and branches for insects. It has a slender, warbler-like beak. Warbler finch, cactus ground finch. Woodpecker, sharp-beaked finch, ground finch. E, B, and BT, small ground. Insectivorous finch, tree finch. Large and medium. Insectee, groundvorous finch, tree finch. Vegetarian, large. Tree ground finch, finch. Figure 8.8. 10 species of Darwin's finches showing differences in bills and feeding habits. The bills of several of these species resemble those of different distinct families of birds on the mainland, a condition that presumably arose when the finches evolved new species in habitats where other kinds of birds normally occur on the mainland and which are not available to finches there. J449. Evolution. To Darwin, it appeared that each was slightly modified from an original colonist, probably the finch on the mainland of South America, some 600 miles to the east. Its adaptive radiation led to the formation of so many species because other birds were few or absent, leaving empty niches to fill, and because the numerous islands of the Galapagos provided ample opportunity for geographic isolation. Types of natural selection. If an individual has some form of a trait that gives him a better chance of survival in a given environment, evidently his offspring inherit the trait. This is known as differential survival. Differential survival would refer to the survival of individuals in an environment to which they are well adapted. The adaptations are genetically controlled and heritable. Differential survival leads to differential reproduction of alleles responsible for the survival of the next generation. Natural selection means differential reproduction among members of the same gene pool. Types of natural selection. 1. Directional selection. 2. Stabilizing selection. 3. Disruptive. Environmental changes don't necessarily induce genetic changes. The genetic variance composing a species may be sufficiently homeostatic so that adaptiveness remains adequate in the altered environments. The Darwinian fitness of variant phenotypes and genotypes may be shifted, some in positive and others in negative directions. A directional natural selection then comes into operation. Transforms the gene pool of a species or population toward the highest level of adaptiveness that can be reached in the new environment. Stabilizing select theory. Extreme varieties from both ends of the frequency distribution are eliminated. The frequency distribution looks exactly as it did in the generation before. A real-life example is that of birth weight of human babies. Features of stabilizing selection _ EQ operate in constant or unchanging environment. Keeps a population genetically constant. E favors the average or normal phenotypes. Introduce homozygosity in the population. O check accumulation of mutation. E selection. Individuals at one end of the frequency distribution do especially well, and so the frequency distribution of the trait in the subsequent generation is the frequency distribution looks exact. Differential survival leads to differential reproduction. Natural selection. If an individual has some form of a trait that gives him a better chance of survival in a given environment, evidently his offspring inherit the trait. This is known as differential survival. Differential survival would refer to the survival of individuals in an environment to which they are well adapted. The adaptations are genetically controlled and heritable. Differential survival leads to differential reproduction of alleles responsible for the survival of the next generation. Natural selection means differential reproduction among members of the same gene pool. Types of natural selection. 1. Directional selection. 2. Stabilizing selection. 3. Disruptive. Environmental changes don't necessarily induce genetic changes. The genetic variance composing a species may be sufficiently homeostatic so that adaptiveness remains adequate in the altered environments. The Darwinian fitness of variant phenotypes and genotypes may be shifted, some in positive and others in negative directions. A directional natural selection then comes into operation. It transforms the gene pool of a species or population toward the highest level of adaptiveness that can be reached in the new environment. Under stabilizing select theory, extreme varieties from both ends of the frequency distribution are eliminated. The frequency distribution looks exactly as it did in the generation before. A real-life example is that of birth weight of human babies. Features of stabilizing selection _ EQ operate in constant or unchanging environment. Keeps a population genetically constant. E favors the average or normal phenotypes. Introduce homozygosity in the population. O check accumulation of mutation. Under direction selection. Individuals at one end of the frequency distribution do especially well, and so the frequency distribution of under direction selection. Individuals at one end of the frequency distribution do especially well, and so the frequency distribution of the trait in the subsequent generation is shifted from where it was in the parental generation. Industrial melanism was such an example. Features of directional selection. Due to the change in the environment in particular direction, favors the phenotype which is non-average or extreme. S alters the mean value of the trait in the population in one direction. Favors accumulation of those mutations that increases fitness in the changing environment. Eliminate normal or average individuals. Sent under diversifying, disruptive selection, both extremes are favored at the expense of intermediate varieties. A clear example is provided by the different color pattern of the butterfly Papilio dardanus. Evolution. Features of disruptive selection. Previously homologous population break up into several different adaptive forms. Extreme values have highest fitness and intermediate or mean values are relatively disadvantageous. Ooeder in a population previously adapted to a non-homologous environment is subjected to divergent selection pressure in different parts of its distributional area. Greater than mean 5. CQ3. O. Original G. Distribution. Phenotype. Directional stabilizing disruptive. Before. Selection at figure 8.9. Directional, stabilizing, and disruptive. Selection. Shaded areas show the groups after being selected against. At the top is the selection original distribution of individuals. The final distributions after selection appear in the bot QM row. 8.5 Pattern of evolution. Convergent evolutio and ND divergent evolution. We often find unrelated animals converging on the same form or structure, because that form is very adaptive in their common environment. This special case of evolution is called convergent evolution. Example of convergent evolution is the streamlined shape of sharks and dolphins. One is a fish, the other is a mammal, and they are related to one another in only the most distant sense. But if your life depends on swift movement through the water, then a streamlined shape is pretty much essential. Another example is wings in birds, bats and flies. Convergent evolution produces analogous structures. Divergent evolution is the process of two or more related species becoming more and more dissimilar. The red fox and the kit fox provide an example of two species that have undergone divergent evolution. Divergent evolution produces homologous structures. The same bones can be used in many ways, leading to several divergent evolutionary paths: frogs, bats, birds, men and so on. Solution. Homoplasy. Demoplasy refers to similarity among unrelated organisms. The wings of birds, bats, and insects are tilde-homoplastic features because the common ancestor of these three groups lacked wings. Eaves and thorns modified stems are an example of homoplasy among plants. Spines and thorns look similar and both provide protection to the plant, but the plants to which they belong are distantly related. Homoplastic similarity can be anatomical, physiological, or behavioral. Coevolution. The term coevolution is used to describe cases where two or more species reciprocally affect each other's evolution. For example, evolutionary change in the morphology of a plant might affect the morphology of a herbivore that eats the plant, which in turn might affect the evolution of the plant, which might affect the evolution of the herbivore, and so on. Evolution is likely to happen when different species have close ecological interactions with one another. These ecological relationships include 1. Predator, prey and parasite, host. 2. Competitive species. 3. Mutualistic species. Plants and insects represent a classic case of coevolution, one that is often, but not always, mutualistic. Many plants and their pollinators are so reliant on one another and their relationships are so exclusive that biologists have good reason to think that the match between the two is the result of a coevolutionary process. But we can see exclusive matches between plants and insects even when pollination is not involved. Some Central American acacia species have hollow thorns and pores at the bases of their leaves that secrete nectar. Species have hollow thorns and pores at the bases of their leaves that secrete nectar. These hollow thorns are the exclusive nest site of some species of ant that drink the nectar. But the ants are not just taking advantage of the plant, they also defend acacia plant against herbivores. This is probably of coevolution. The plants would not have evolved hollow thorns or nectar pores unless their evolution had been affected by the ants, and the ants would not have evolved herbivore defense behaviors unless their evolution had been affected by the plants. Parallel evolution. Evolution is the development of similarities in separate but related evolutionary lineages through the operation of similar selective factors on both lines. Parallel evolution is similar to convergent evolution in that similar adaptations arise in different species. The difference is that in parietal evolution the two species have a relatively recent common ancestor. Adaptive radiation. Natural selection can ultimately lead to the formation of new species. Sometimes many species evolve from a single ancestral species. Similarities in skeletal and muscular structure of Hawaiian honeycreepers fed scientists to conclude that the 23 species of honeycreepers evolved from one ancestral species. Such an evolutionary pattern, in which many related species evolved from a single ancestral species, is called adaptive radiation. Adaptive radiation most commonly occurs when a species of organism successfully invades an isolated region where few competing species exist. If new habitats are available, new species will evolve. For example, there are 14 species of finches on the Galapagos Islands, each filling a different niche on different islands. All of them evolved from one ancestral species, which colonized the islands only a few million years ago. This process, whereby species evolve rapidly to exploit empty ecospace, is also present an example of adaptive radiation. ARYD. Evolution. 8.6 Species and speciation. Concept of species. Biological species concept. The biological species concept put forward by Mayer defines a species as members of populations that actually or potentially interbreed in nature, not according to similarity of appearance. Although appearance is helpful in identifying species, it does not define species. This concept emphasizes that a species is an evolutionary unit. Members share genes with other members of their species, and not with members of other species. Phenetic species concept. A species is a set of organisms that are phenotypically similar and that look different from other sets of organisms. According to this concept, phenotypic similarity is all that matters in recognizing separate species. Example: if the two frogs look same, even though they are prevented from mating with each other, they would be considered the same species according to the phonetic species concept. Evolutionary phylogenetic species concept. An evolutionary phylogenetic species is a lineage and ancestral descendant sequence of populations evolving separately from others and with its own evolutionary role and tendencies. Evolutionary lineage is often conveniently represented in a tree-like diagram called a cladogram. Construction of evolutionary trees is termed cladistics. Cladistics. Cladistics is the classification of species based on phylogeny or evolutionary ancestry. The term phylogenetics is often used synonymously with cladistics. Cladistics is distinguished from other taxonomic systems because it focuses on the evolutionary relationships of species rather than on morphological similarities. Cladistics generates diagrams called cladograms that represent the evolutionary tree of life. Evolutionary tree is represented by lines and nodes. Its species are sequences of DNA, proteins which are compared or located at external nodes but are connected via branches to interior nodes which represent ancestral forms for two or more species or sequences. The lengths of the branches indicate the degree of difference between the sequence species represented by the nodes. CBA less than, external node species D species C species B species A. Branch. Internal node, speciation event. An evolutionary tree and evolutionary species tree. The terms evolutionary tree, and sometimes phylogenetic tree are often used synonymously with cladogram. However, phylogenetic tree and cladogram is not same. A phylogenetic tree is a specific type of cladogram where the branch lengths are proportional to the predicted evolutionary time between organisms or sequences. Pseudograms are branch diagrams, similar in appearance to family trees, that illustrate patterns. Evolution. Relatedness where the branch lengths are not necessarily proportional to the evolutionary time between related organisms or sequences. Understanding a phylogeny is a lot like reading a family tree. The root of the tree represents the ancestral lineage, and the tips of the branches represent the descendants of that ancestor. As you move from the root to the tips, you are moving forward in time. When a lineage splits, speciation, it is represented as branching on a phylogeny. When a spuathlon event occurs, a single ancestral lineage gives rise to two or more daughter lineages. Descendants, species D, species C, species B, species A. Recent 1. Plus, ancestor past and evolutionary species tree. In the above figure present products of the speciation processes, where one species becomes subdivided into two or more groups that follow their own evolutionary ways to become new species, coexisting at the same time. Certainly different branches of a cladogram reflect the idea that these populations are considered to have evolved separately with their own evolutionary roles and tendencies. An evolutionary tree may be rooted or unrooted. A rooted tree infers the existence of a common ancestor and indicates the direction on evolutionary process. An unrooted tree does not infer a common ancestor and shows only the evolutionary relationships between the organisms. Rooted tree, unrooted tree. A group of species derived from a single common ancestral population is said to have a monophyletic origin. Thus, a monophyletic group is a clade, consisting of an ancestor and all its descendants. A clade is defined as a group consisting of a single common ancestor and AF its descendants. A group of organisms is said to be paraphyletic if the group contains its most recent common ancestor but does not contain alpha descendants of that ancestor. Thus a paraphyletic group is a monophyletic group from which one or more of the clades is excluded to form a separate group. A group that does not contain the most recent common ancestor of its members is said to be polyphyletic. Example, the group consisting of warm-blooded animals is polyphyletic, because it contains both mammals and birds, but the most recent common ancestor of mammals and birds was cold-blooded. Era. Evolution. LTEN. 2S right, high PT street, asterisk, underscore underscore, underscore underscore. LOL0Y, SOL2 NKGIYNGE, Y3 cents, NNEY 532%. QSN and YY5 Go 9 YV0S section and 3 RSS square S0 OB QYH, E. RI section $% section H and A29 LAR and ARA, RA and RA and ASL and N pound FSG and QSYR. A. Evolution. Evolution. LTEN. 2S. Dash. Reptilia. Amniota. Tilde. Amniota. Tetrapoda tetrapoda tetrapoda. Monophyletic paraphyletic. Speciation is a lineage splitting event that produces two or more separate species. Gene flow is reduced sufficiently between sister populations to allow each to become irrevocably committed to different evolutionary lineages. It occurs when gene flow is reduced sufficiently between sister populations to allow each to become irrevocably committed to different evolutionary lineages. Imagine that you are looking at a tip of the tree of life that constitutes a species of fruit fly. Move down the phylogeny to where your fruit fly twig is connected to the rest of the tree. That branching point, and every other branching point on the tree, is a speciation event. At that point, genetic changes resulted in two separate fruit fly lineages, where previously there had just been one lineage. But why and how did it happen? Diventissima di Melanogaster di Obscura. Figure 8.10. Diagram presenting speciation. Event in genus Drosophila. Speciation events. Anagenesis versus cladogenesis. Most evolution consists of two processes, anagenesis, also known as phyletic speciation, and cladogenesis. Phyletic evolution is the progressive evolution of species involving a gradual change in gene frequency in an entire population. When enough mutations reach fixation in a population to significantly differentiate from an ancestral population, a new species name may be assigned. That the entire population is different from the ancestral population so that the ancestral population can be considered extinct. A species is transformed over time by the acquisition of phenotypic and genetic modification. There is no increase in the number of species. Speciation results in the splitting of a lineage into two or more species as described as cladogenesis that subsequently diverge in their traits through anagenesis. Evolution. Species 2, Species 2, Species 3. Figure 8.11. Forms of speciation. In anagenesis, a species changes over time until it is so term different from its progenitor that it is classified as a new species. In cladogenesis, B. Speciation takes as a branching. Species 1 process wherein one species becomes two or more. A, B. Examples of speciation. Diane Dodd and her colleagues studied the evolution of digestive physiology in Drosophila pseudoobscura. They collected flies from a wild population and established several separate laboratory populations. Some of the laboratory populations were raised on starch-based medium and others on maltose-based medium. Both food sources were stressful for the flies. It was only after several months of evolution by natural selection that the populations become adapted to their artificial diets and began to thrive. Dodd to know whether the starch-adapted populations and the maltose-adapted populations, which diverged under strong selection and in the absence of gene flow, had become different species. Roughly a year after the populations were established, a series of mating trials were performed. For each trial, 48 flies were placed in a bottle, 12 males and 12 females from a starch-adapted population and 12 males and 12 females from a maltose-adapted population. The researchers then noted which flies mated. Dodd predicted that if the populations adapted to different media have speciated, then the flies would prefer to mate with members of their own population. If the populations had not speciated, then the flies would mate at random. Rougie 600 of the 900 matings observed were between males and females from the same population. In other words, the differently adapted fly populations showed partial pre-mating isolation. Dodd and her colleagues concluded that the populations had begun to speciate, but had not yet completed the process. Incipient speciation in laboratory populations of Drosophila pseudoobscura, number of matings involving each kind of male-female pair. Female. Starch-adapted maltose-adapted. Starch-adapted 290,153 maltose-adapted 149,312. Source. Compiled from Dodd, DMB 1989. Reproductive isolation is a consequence of adaptive divergence in Drosophila pseudoobscura. Evolution. Mechanism of cladogenesis. Reproductive isolation. There are many barriers to reproduction that evolve during the course of speciation which limit gene flow between sister species. Each species may have its own courtship displays, or breeding season, so that members of the two species do not have the opportunity to interbreed. Or, the two species may be unable to interbreed successfully because of failure of the egg to become fertilized or to develop. This is full dichotomy between pre-mating or prezygotic, i.e., pre-zygote formation, reproductive isolating mechanisms, and post-mating or post-zygotic isolating mechanisms. Evolution. Prezygotic isolating mechanisms. Prezygotic isolating mechanisms lead to reproductive isolation by preventing the formation of hybrid zygotes. 1. Ecological isolation. Species occupy different habitats. 2. Temporal isolation. Species breed at different times. 3. Behavioral isolation. Species engage in distinct courtship and mating rituals. 4. Mechanical isolation. Interbreeding is prevented by structural or molecular blockage of the formation of the zygote. Mechanisms include the inability of the sperm to bind to the egg in animals, or the female reproductive organ of a plant preventing the wrong pollinator from landing. Zygotic isolating mechanisms. Post-zygotic mechanisms are those in which hybrid zygotes fail, develop abnormally, or cannot self-reproduce and establish viable populations in nature. 1. Hybrid inviability. Development of the zygote proceeds normally but the hybrid does not survive. 2. Hybrid sterophyty. The hybrid is healthy but sterile. The mule, the hybrid offspring of a donkey and a mare, is sterile, it is unable to produce viable offspring. 3. Hybrid is healthy and fertile, but less fit, or infertility appears in later generations, as witnessed in laboratory crosses of fruit flies, where the offspring of second-generation hybrids are weak and usually cannot produce viable offspring. Allopatric, PARAPATRIC, and sympatric speciation. Reproductive ISD-latting mechanisms are barriers to gene flow, the spread of genes between populations. These isolating mechanisms can evolve in 3 different ways, each of which defines a different mechanism of speciation. In 1963, Ernst Mayr contended that speciation takes place through allopatry, or geographic isolation, allopatric speciation. Allopatric speciation is genetic divergence permitted by geographic isolation. This process is dependent on random mutation, which accumulates steadily after a population has been subdivided in space. This subdivision can take place because of geographic barriers such as mountain ranges and water bodies. Because the 2 subpopulations are then reproductively isolated, each accumulates different mutations and the 2 populations diverge. 1. Allopatric parapatric sympatric. Original. Population. Initial step, figure, 8.12. Of speciation, the 3 general mechanisms of barrier, new niche genetic speciation. In allopatric speciation, formation entered polymorphism, reproductive isolation evolves after the population has been geographically evolution of reproductive isolated. In parapatric speciation, reproductive evolution. This divergence is strongest where the population size of one or both of the subpopulations is small because sampling error is greatest in smaller populations. These differences will eventually lead to reproductive incompatibilities that will keeping the populations distinct. One well-known example of allopatric speciation involves divergence of Drosophila populations in the Hawaiian archipelago. Reproductive isolating mechanisms usually originate incidentally in the speciation process. That is, they arise incidentally during the process of evolution in isolated populations rather than being selected for. When isolated populations come together again, incomplete isolating mechanisms may allow hybrids to form. If the hybrids are normal and viable and can freely interbreed with individuals of each parent population, then no speciation has taken place. However, if the hybrids are at a disadvantage, natural selection may favor stronger isolating mechanisms. In this case, organisms that mate with individuals from the other population leave fewer offspring. The result is a more effective barrier to hybridization. Regions in which previously isolated populations come into contact and produce hybrids are called hybrid zones. Sympatric speciation, conversely, does not require geographic isolation. Instead, it relies on the development of reproductive isolation mechanisms to allow divergence of the 2 subpopulations. This reproductive isolation can be a result of a number of factors, including competition for resources, disruptive selection, and sexual selection. In sympatric speciation, selection acts against individuals of an intermediate type, either through decreased viability or decreased fecundity. This creates the evolution of mating preferences and other mechanisms that result in decreased hybridization. Sympatric speciation, selection enhances traits that promote the divergence of the populations. Fundamentally different from allopatric speciation, where mutation passively leads to differences that allow divergence of the populations. Probably the best known example of sympatric speciation is the divergence of Rhagoletis formonia, the maggot fly. This has recently diverged into 2 subspecies due to the introduction of apple trees in the northeastern United States, where hawthorn trees were native. Initially, the flies used the hawthorn fruits to reproduce on and to lay their eggs. The introduction of the apple trees provided a more nutritious food source for developing maggots, as well as an escape from parasitic wasps. Between the 2 types of fruits, such as maturation timing, allowed for the evolution of escalation mechanisms and subsequent divergence of the 2 subspecies. Parapatrication is a form of speciation in which the evolution of reproductive isolating mechanisms occurs when a population enters a new niche or habitat within the range of the parent species. Generally, this occurs when there has been a drastic change to the environment within the original species habitat. In parapatric speciation, there is no specific extrinsic barrier to gene flow. The population is continuous, but nonetheless, the population does not mate randomly. Individuals are more likely to mate with their geographic neighbors than with individuals in a different part of the population's range. In this mode, divergence may happen because of reduced gene flow within the population and varying selection pressures across the population's range. 8.7 Evolutionary forces involved in speciation. Genetic change lies at the heart of speciation. The formation of new species involves the action of some evolutionary forces known to cause genetic changes within populations. Principal evolutionary forces most commonly involved in species divergence are natural selection, genetic drift, sexual selection, and mutation. All these forces act to change allele frequencies within populations over time, and there is no question that they operate during speciation as well. 1. Natural selection. Darwin first raised the idea that speciation could readily occur through the prolonged action of natural selection, the process by which genetic variants that are better suited to the natural environment increase in frequency while variants that are not well suited decrease, described earlier. R. L. 2. 3. Evolution. Genetic drift.